Animals, no cell is an island. They communicate between themselves with a variety of molecules. Many unicellular organisms respond to external signaling molecules, and the signaling reaches the highest level of multicellular organisms. External signaling molecules mostly interact with their specific receptor molecules on the membrane of the target cells, Big 4.1, and thus membrane receptors constitute the largest variety in signaling pathways. For cell communication and cell signaling, biosignaling, GPCR signaling. Stimulus and response are coupled in all living organisms, and the phenomenon that links these two is cell signaling. Signaling could be intracellular or extracellular, but it always serves to bridge the gap between a stimulus from the environment or from within itself and its corresponding response. Like all social. Figure 4.1 schematic diagram depicting a generalized membrane receptor signaling pathway. The figure shows 1. A signaling cell is secreting an extracellular signaling molecule or ligand, also known as the first messenger, which then binds to a transmembrane receptor. 2. Activation of the receptor upon ligand binding might cause changes in the bound proteins 3 or in the cytosolic domain of the receptor 4a. This may lead to the production of second messenger molecules 5 and subsequent attachment of adapter or effector proteins 6. This will lead to the activation of a set of proteins 7 and, upon cascade activation, 8 will ultimately cause several cellular changes 9. GPCR structure and function. In 1994, Alfred G. Gilman and Martin Rabo were awarded the Nobel Prize in Physiology or Medicine for their discovery of G proteins and the role of these proteins in signal transduction in cells. 18 years later, Brian K. Kubilka and Robert J. Lefkowitz won the Nobel Prize in Chemistry for studies of G protein coupled receptors (GPCRs). G protein coupled receptor (GPCR), also known as 7-transmembrane domain receptor, 7-TM receptor, heptahelical receptor, serpentine receptor, and G protein link receptor, is a crucial eukaryotic signal transduction gatekeeper that senses molecules outside the cell and activates inside signal transduction pathways and ultimately cellular responses to the external stimuli. The GPCR superfamily is the largest single family of proteins in the human proteome consisting of approximately 1,000 members, encoded by at least 831 different human genes, which amount to about 4% of the human protein coding genome, based on genome sequence analysis. Based on the similarities in the transmembrane domain, these have been classified into 5 subfamilies: Rhodopsin family 701 members, adhesion family 33 members, frizzled taste family 24 members, glutamate family 15 members, and secretin family 15 members. 1. Rhodopsin family. This is the largest subfamily accounting for approximately 80% of all GPCRs, with receptors for a variety of ligands, including nucleotides like CAMP, lipids, and small peptides including pheromones, hormones, neurotransmitters, biogenic amines, and light. These generally contain a short 35 residue N-terminal tail for biogenic amines, with exceptions for glycoprotein receptors which contain approximately 350 residue leucine-rich domain for hormone binding. Rhodopsin-like GPCRs have been divided into 21 subgroups based on a phylogenetic analysis according to the HGNC database. 2. Adhesion family. The second largest GPCR family is the adhesion family, with 33 members. This family is very special due to its members' secondary structures, with distinctive long N-termini containing a number of adhesion domains that facilitate cell matrix interactions, as is prevalent in cell adhesion molecules like integrins, selectins, and cadherins. These studies have shown that adhesion GPCRs are involved in the signaling of cell adhesion, motility, immune system and embryonic development, neuronal development, hematopoietic systems, and cancer, especially cancer progression and metastasis in cases of melanoma, breast, lung, and gastric cancer. 3. Frizzled taste family. These members comprise frizzled and taste receptors. While the former is involved in WNT and other signaling pathways and has been implicated in establishing and controlling planar cell polarity, embryonic development, cell proliferation, formation of neural synapses, and many other processes in developing in adult organisms, the latter facilitates the sense of taste. There are 11 genes in frizzled receptors, including FZD1, FZD2, FZD3, FZD4, FZD5, FZD6, FZD7, FZD8, FZD9, FZD10, and SMO apart from the taste receptor genes. 4. Glutamate family. Metallotropic glutamate receptors have a large extracellular N-terminus which responds to the orthosteric endogenous ligand. They are coupled to G proteins and stimulate the inositol phosphate CA2+ intracellular signaling pathway and function mainly in neurotransmission. There are 4 subgroups, including calcium sensing receptors, gamma-aminobutyric acid B receptors, GABDR, glutamate receptors, metallotropic, GRM, and class C orphans. 5. Secretin family. Secretin-like GPCRs have been divided into 6 subgroups based on a phylogenetic analysis according to the HGNC database, including adhesion G protein coupled receptors, ADGR, calcitonin receptors, CALCR, corticotropin releasing factor receptors, CRHR, glucagon receptor family, vasoactive intestinal peptide receptor family, and parathyroid hormone receptors. Members of this family are generally regulated by peptide hormones like secretin, glucagon, parathyroid hormones, etc. 6. Orphan GPCRs. All other GPCRs without known functions have been classified as orphan GPCRs. The GPCRs have 7 transmembrane alpha helices with a C-terminal end inward and the N-terminal projected outward. They are characterized by 3 extracellular loops L1 to L3 and 3 intracellular loops L1 to L3 all hydrophilic. Each transmembrane domain consists of approximately 20 to 30 hydrophobic amino acid residues and is long enough, approximately 30 nanometers. The extracellular parts of the receptor can be glycosylated. These extracellular loops also contain 2 highly conserved cysteine residues that form disulfide bonds with transmembrane helices to stabilize the receptor structure. The GPCR arranges itself into a tertiary structure resembling a barrel, with a set of transmembrane helices forming a cavity within the plasma membrane that serves as a ligand binding domain that is often covered by the second extracellular loop. Ligands may also interact with extracellular loops in the N-terminal tail. The N-glycosylation motif in the extracellular part takes part in receptor folding, trafficking, etc. Palmitoylation is the intracellular part after helix 8 is essential for lipid raft localization. The DRY motif in the second intracellular loop takes part in receptor activity and protein-protein interactions, Big 4.2. Desensitization of the receptor occurs through the 3 motifs, PKC phosphorylation, GRK phosphorylation, and PKA phosphorylation. Figure 4.2 topological structure of GPCRs. This is a schematic diagram of the structure of an ideal GPCR and the positions of its various alpha helical loops and motifs topologically in the plasma membrane. The side view on the left shows the heptahelical nature with positions of different motifs and chemical groups, while the top view illustrates the relative position of helices. Ligand binding and activation. The ligands for GPCRs range from photon or subatomic particles to ions H+ and Ca2+ to small organic molecules to peptides and proteins and transmit the resulting signals across the plasma membrane over a distance of approximately 30 A. Small organic molecules generally bind within the transmembrane segments, and peptide hormones and proteins often bind to the amino terminus and extracellular sequences joining the transmembrane domains. High-resolution crystal structures and biochemical studies have revealed that the nucleotide binding pocket of G protein is located between the Ross-like domain and the alpha helical domain of G alpha subunit, surrounded by 4 flexible regions P loop, switch I, switch 2, and switch 3. The N-terminus of G alpha subunit is reported to be critical for the structure and function of G alpha subunit and is Myers-related or palmitoylated, suggesting the role of this region in the attachment to the plasma membrane. Most studies reveal that the C-terminus of G alpha subunit is inserted into the cytoplasmic pocket surrounded by TM domains with possible interaction with TM6-ICL3. The alpha and beta 1 hinge region of the G alpha subunit also contributes in translating receptor-induced conformational changes to the nucleotide binding pocket as the beta 1 sheet is connected to the P loop that interacts with the phosphate group of GDP. Ligand binding to the GPCR results in the formation of a ligand receptor complex. This results in the interaction of the cytoplasmic third and fourth C3 and C4 loops with the G protein. The release of bound GDP from the inactive G protein is mediated by guanine nucleotide exchange factor GDF. Now binding of GTP in place of GDP is favored by the high cellular concentration relative to its binding affinity. This induces a conformational change in at least 2 highly conserved segments of the protein, switch I and switch 2, allowing the protein to bind to GTP. In the active state, switch I and switch 2 are bound to the terminal Y-phosphate of GTP through interactions with the backbone amide groups of conserved threonine 35 and glycine 60 residues. The Ross-like domain hydrolyzes GTP and provides binding sites for the G beta subunit. However, the proper mechanism of activation of G protein by GPCR upon ligand binding is still under investigation. Different classes of ligands and their examples are tabulated below. Classes ligands. Neurotransmitters epinephrine, norepinephrine, dopamine, 5-hydroxytryptamine, histamine, acetylcholine. Adenosine, opioids. Catechinase substance P, neuropilin A, neuropeptide K. Other peptides angiotensin 2, arginine vasopressin, oxytocin, VIP, GRP. TRH, PTH glycoprotein hormones TSH, FSH, LH, HCG. Arachidonic acid derivatives thromboxane E2. Others odorants, tastans, endothelins, platelet activating factor, cannabinoids, light. Table 4.1 examples of ligands for GPCRs. Signal amplification. There happen to be a few thousand receptors on the cell membrane of each cell, and on the production of ligand molecules which too is minuscule concerning the response generated, there is always a need for substantial amplification of the ligand responses. Signaling cascades have long been thought to amplify input signals, as each molecule in the cascade can serially activate multiple molecules of a downstream component of the cascade. For example, in the case of the epinephrine hormone signaling cascade, the response needs to be fast to correspond to the flight or fight response. Upon production of epinephrine by the gland, it reaches target tissue with specific receptors. Because the concentration of the hormone in the blood is normally very low, and LT 10 to 10 m, the signal has to be amplified. Upon ligand binding, a conformational change activates a resting heterotrimeric G protein associated with the inner leaflet of the membrane. Activation of a single receptor can result in even more than 10 active heterotrimeric G proteins, providing a great boost to the transduction of the first messenger and a good deal of amplification of signal big 4.3. The binding of a single ligand stimulates a large number of adenyl cyclase, and each of the activated adenyl cyclase produces a large number of camps. Now each of the two camps activates a pKa that in turn phosphorylates several other proteins and so on, thus rapidly amplifying the external signal. Figure 4.3 schematic depiction of the amplification of signals by signaling pathways in several orders of messengers. The above chart depicts the hierarchical structure of the camp pKa signaling pathway and the distribution of amplifying signals at each level having the ligand epinephrine, adenyl cyclase, camp, pKa, pKa activated enzymes, and ultimately the products of those enzymes. The concentrations of certain molecules in the cell are mentioned, like the ligand epinephrine is 10 to 10 m, which in turn increases to 10 to 6 m per camp. Weak and strong agonists. In simple terms, agonists are chemicals that bind to a specific receptor and induce certain conformational changes to activate the receptor. The agonists and antagonists of GPCRs have been discovered in large numbers, and their application of them has largely been limited to structural studies, including activation of GPCRs and relaying the signal to G proteins. There could be several agonists for a specific receptor and vice versa. In a separate approach, a lot of mimetic synthetic agonists have been made for specific receptors to study their functions in greater detail. GPCRs being a large family, these receptors have an enormous number of agonists. For contra, weaker partial agonists are antagonists who elicit the same response as the agonists but to a lesser extent. These compounds possess low intrinsic activity, thus having partial efficacy in biological responses compared to a strong agonist. Antagonists. Antagonists act opposite to the agonists and reverse the biological response initiated by agonists. GPCRs also have a plethora of different antagonists specific to receptors. Antagonists are either naturally occurring compounds or molecules present in the body or some chemically designed artificial ones with therapeutic significance. Adenyl cyclase. Adenyl cyclase AC is a protein embedded in the lipid bilayer of the cell membrane. This enzyme catalyzes the conversion of ATP to CAMP, being stimulated by G alpha S subunit. This activation is very essential regarding the broad array of signals that can be transduced by CAMP intracellularly. AC is an integral membrane glycoprotein present as a single polypeptide possessing 12 transmembrane domains. It has an approximate molecular size of 115 to 150 kDa. AC in humans is encoded by 9 different genes and 10 isoforms of this enzyme have been described. In mammals, there are 9 membrane-bound AC1 to 9 and 1 soluble adenyl cyclase SAC that generate CAMP based on their distinct regulation and expression patterns, determined by G by subunits, CA2 plus calmodulin, kinases, and NO. For example, G by subunits stimulate AC2 and 4 while suppressing AC1, 5 and 6. CA2 plus calmodulin stimulates AC1, 3 and 8, while inhibiting AC5 and 6. PKC stimulates all acts except AC6, which is inhibited. The membrane-bound enzyme contains 2 similar catalytic domains on the cytosolic phase of the membrane and 2 integral membrane domains, each of which is thought to contain 6 transmembrane alpha helices, big 4.4. The binding of a ligand to the stimulatory RS receptor or inhibitory RI receptor results in receptor-mediated activation of G protein, which entails the exchange of GDP by fresh GTP molecule on alpha subunit and the concomitant dissociation of bicomplex from alpha. The alpha subunit of stimulatory G protein has 2 helices turned to switch I and switch 2. 2 regions of the alpha subunit, i.e., G alpha S. GTP, the helix in switch 2 and the alpha 3 beta 5 loop interact simultaneously with a specific active site domain of adenyl cyclase, activating it. Activation of it then results in augmented enzyme activity, rapidly converting ATP to CAMP. There is also microheterogeneity of adenyl cyclase, although generally, a single enzyme has been assumed to exist for all cyclic receptors. AC1 is important for its role in learning and memory processes, and its activation enhances memory formation. AC3 has a role in peripheral and behavioral anosmia. AC6 overexpression on cardiac cues improves cardiac function. Certain experiments also suggest the bronchodilatative and immunomodulatory role of AC9 in the lungs. Soluble AC SAC is mainly expressed in the testes, and any experimental inhibition of SAC causes the inability of the mouse sperm to undergo capacitation. Inhibitors of AX include PMC6, SQ22536, HTSO9836, NKY80, KH7, etc. Forskolin and Culver-syndromic hydrochloride are known to activate most AX. Figure 4.4 structure of adenyl cyclase. This is a pictorial representation of the generalized structure of adenyl cyclase, showing the presence of both NH2 and COOH groups on the intercellular side, as well as containing ATP binding, MG2 plus binding and forskolin binding sites. Receptor tyrosine kinases, RTKs. An enzyme link receptor or catalytic receptor is a transmembrane receptor where the binding of an extracellular ligand causes enzymatic activity on the intercellular side. Hence these receptors are integral membrane proteins possessing both enzymatic catalytic and receptor functions. The ligands for RTK include insulin, epidermal growth factor EGF, transforming growth factor alpha TGF alpha, platelet-derived growth factor PDGF, fibroblast growth factor FGF, and choline stimulating factor 1 CSF1. Examples of RTK are the ERB family, GDNF, glial cell-derived neurotrophic factor family, NPR, natriuretic peptide receptor, TRK, neurotrophin receptor, and toll-like receptors, etc., among others. The activated RTKs may interact with several types of proteins, including GTPase activating protein, GAP phospholipase CY, and the SRC non-receptor protein tyrosine kinases. Figure 4.5 structure and activation of RTKs. The structure of a single RTK is outlined in the diagram. The next 2 insets towards the right show the recruitment of another RTK on ligand binding and subsequent autophosphorylation of tyrosine residues for activation of the receptors. A very common example of the RTK pathway is the insulin signaling pathway. The insulin receptor is an RTK which consists of 2 alpha subunits on the outer face of the plasma membrane and 2 transmembrane beta subunits. The binding of insulin to the alpha subunit triggers a conformational change that allows the autophosphorylation of the tyrosine residues in the COOH domains of the beta subunits. Multidocking proteins like insulin receptor substrate 1 IRS1 then binds one of the phosphotyrosine residues via PTB domain. The activated RTK then phosphorylates the bound docking protein, forming some phosphotyrosine residues, which in turn will serve to recruit SH2 containing signaling proteins, thus amplifying the signal at the receptor level. Pi3 kinase is an enzyme bound to the inner leaflet of the plasma membrane, having 9 homologs encoded by the human genome. Pi3 kinase is made up of 2 proteins named P85 with an SH2 phosphotyrosine binding domain and P110 subunit with catalytic activity. This recruitment of Pi3 kinase to the plasma membrane brings it near the sphenonucleotide substrates, leading to the formation of Pi3, 4-bisphosphate or Pi3, 4,5-triphosphate. Now AKT or protein kinase B PKB is a S or stress-specific protein kinase that plays a key role in multiple cellular processes. AKT possesses a plextrin homology pH domain that binds to the 3-phosphate group of Pi3, 4-bisphosphate. The PTK1 kinase also binds to the 3-phosphate groups to get activated. The other 2 domains of AKT are kinase and regulatory domains, where PTK1 phosphorylates a threonine 308 residue in the kinase domain, and PTK2 and ALK is assigned to a serine 473 residue in the regulatory domain to render the protein to be fully active. Activation of AKT takes 5 to 10 minutes, but its effects last as long as several hours. In case of insulin signaling, AKT phosphorylates RGC2, ROL gap complex, and AS160 AKT substrate of 160 kDa in the GSP, which is required in the insulin-mediated translocation of the glucose transporter BLEP4 to the plasma membrane. Besides increasing the exocytosis of BLEP4, AS160 inhibits its endocytosis via downstream target RAP10 and adipocytes. This facilitates the uptake of glucose from the blood, thereby bringing about the hypoglycemic effect, Fig 4.6. Figure 4.6 RTK signaling with the insulin hormone as ligand. This diagram depicts the exact signaling interplay between RTK pathway members that takes place in response to insulin culminating in the localization of BLEP4 transporters to the cell membrane. When insulin molecules bind to the RTKs, there occur phosphorylations in the intracellular domain of the receptor, where the insulin receptor substrate IRS binds. IRS facilitates the recruitment of Pi3 kinase in close vicinity to PIP2, which is converted to PIP3 by Pi3 kinase. Now with the help of 2 different enzymes, PTK1 and PTK2, AKT or protein kinase B is activated in proximity with PIP3. AKT next phosphorylates certain components of the BLEP4 vesicle, causing translocation of the vesicles to the cell membrane, thus facilitating glucose uptake by them. Signaling pathways. AKT signaling pathway. Introduction. Since its discovery 30 years ago, from clone versus AKT oncogene after retrovirus transformed with AKT8, many other labs cloned and characterized the 57 kDASR and THR protein kinase, the cellular homologue of versus AKT. Three isoforms of AKT-PTB are conserved in the mammalian genome: AKT1 PKBA, AKT2 PKBB, and AKT3 PKBG. AKT has been identified as a squamous type 3 kinase PI3K, effector molecule and acts downstream to PI3K, a lipid kinase that acts in response to insulin growth factors. Mechanism and regulation. Class 1 PI3K phosphorylates the inositol group of the squamositides, resulting in lipid second messengers PTDINS3, 4P2Pi3, 4P2 and PTDIN3, 4, 5P3KIP3. These serve as a docking site for membrane localization of cluster homology pH domains containing molecules like AKT and PDK1. Once phosphorylated, AKT regulates various cellular functions downstream, like cell survival, proliferation, metabolism, cellular growth, and many others, by either activating or inhibiting molecules like FOXOs, BAD, GSK3, MDM2, TSC2, caspase 9, AS160, PROS40, and ENOS by phosphorylation. Phosphorylation of FOXO3 leads to survival signaling, and its overactivation can induce cell death by apoptosis. AKT inhibits GSK3 beta protein by phosphorylating its subunits, resulting in dephosphorylation of glycogen synthase and an increase in glycogen synthesis. Concomitant to GSK3 beta discovery, which is an AKT substrate from insulin-stimulated cells, it led to the finding of hundreds of other AKT substrates associated with various cellular functions and diseases. Physiological significance. AKT signaling pathway has been reportedly observed to influence various disease progression and shown to be a promising therapeutic target as such or in combination, for example, in case of solid tumors. Allosteric antagonist MK2206 has been shown to inhibit AKT kinase activity in a dose-dependent manner in breast, melanoma, and lung cancer. MK2206 showed to have greater inhibition as compared to other ATP-competitive inhibitors due to the fact that allosteric inhibitors can lock kinase activity in close conformation. Using AKT inhibitors as a single therapeutic agent may not be effective in the long term as inhibition of AKT kinase leads to induction and phosphorylation of other receptor tyrosine kinases, which in turn can activate PI3K by some other non-AKT targets downstream. Preclinical data obtained from MK2206 in combination with anti-estrogen therapy showed to be more efficacious in controlling breast cancer cell proliferation. Besides activation by external stimuli, the AKT signaling pathway is also regulated by phosphatase P10, which inhibits AKT activity. P10 tumor suppressor is a prime target of inactivation in somatic cancers. The enzymatic action of P10 exerts an opposing effect of PI3K, and thus leads to the accumulation of PIP3. Figure 4.7 AKT network and downstream signaling for different cellular activities. Once phosphorylated, AKT regulates various cellular functions downstream like cell survival, proliferation, metabolism, cellular growth, and many others by either activating or inhibiting molecules like FOXOs, BAD, GSK3, MDM2, TSC2, caspase 9, AS160, PROS40, and ENOS by phosphorylating. c-Met signaling pathway introduction. Receptor tyrosine kinase c-Met and its ligand hepatocyte growth factor HGF have been reported to be associated with various types of malignancies. Proto-oncogene c-Met is located on chromosome 7q21 to 31. c-Met is expressed in epithelial cells of various organs like kidneys, muscles, pancreas, liver, and bone marrow. HGF secreted by mesenchymal cells has a pleiotropic effect as well as cytokine and influences cellular function by promoting cell survival, proliferation, scattering, motility, and morphogenesis. It also plays a very important protective function in various disease conditions like liver cirrhosis, progressive neuropathies, and so on. Mechanism and regulation. The inner side of the c-Met comprises a tyrosine kinase catalytic domain bordered by juxtamembrane and carboxy terminal sequences. c-Met-mediated cell scattering involves disruption of cell-cell contact by cadherin-associated changes leading to cell motility, which is a vital process in embryogenesis and wound repair. HGF binding to CMET results in cascade of events downstream, which initiates from c-Met receptor homodimerization and subsequent phosphorylation of tyrosine residues Y1234 and Y1235 located within the catalytic loop of the kinase. Followed by this, the carboxyterminal tail becomes phosphorylated, which then recruit adaptor proteins or signaling effectors. Rhodopsin family. This is the largest subfamily accounting for approximately 80% of all GPCRs, with receptors for a variety of ligands, including nucleotides like CAMP, lipids, and small peptides including pheromones, hormones, neurotransmitters, biogenic amines, and light. These generally contain a short 35 residue N-terminal tail for biogenic amines, with exceptions for glycoprotein receptors which contain an approximately 350 residue leucine-rich domain for hormone binding. Rhodopsin-like GPCRs have been divided into 21 subgroups based on a phylogenetic analysis according to the HGNC database. 2. Adhesion family. The second largest GPCR family is the adhesion family, with 33 members. This family is very special due to its members' secondary structures, with distinctive long N-termini containing a number of adhesion domains that facilitate cell matrix interactions, as is prevalent in cell adhesion molecules like integrins, selectins, and cadherins. These studies have shown that adhesion GPCRs are involved in the signaling of cell adhesion, motility, immune system and embryonic development, neuronal development, hematopoietic systems, and cancer, especially cancer progression and metastasis in cases of melanoma, breast, lung, and gastric cancer. 3. Frizzled taste family. These members comprise frizzled and taste receptors. While the former is involved in WNT and other signaling pathways and has been implicated in establishing and controlling planar cell polarity, embryonic development, cell proliferation, formation of neural synapses, and many other processes in developing in adult organisms, the latter facilitates the sense of taste. There are 11 genes in frizzled receptors, including FZD1, FZD2, FZD3, FZD4, FZD5, FZD6, FZD7, FZD8, FZD9, FZD10, and SMO apart from the taste receptor genes. 4. Glutamate family. Metallotropic glutamate receptors have a large extracellular N-terminus which responds to the orthosteric endogenous ligand. They are coupled to G proteins and stimulate the inositol phosphate CA2+ intracellular signaling pathway and function mainly in neurotransmission. There are 4 subgroups, including calcium sensing receptors, gamma-aminobutyric acid B receptors, GABDR, glutamate receptors, metallotropic, GRM, and class C organs. 5. Secretin family. Secretin-like GPCRs have been divided into 6 subgroups based on a phylogenetic analysis according to the HGNC database, including adhesion G protein coupled receptors, ADGR, calcitonin receptors, CALCR, corticotropin releasing factor receptors, CRHR, glucagon receptor family, vasoactive intestinal peptide receptor family, and parathyroid hormone receptors. Members of this family are generally regulated by peptide hormones like secretin, glucagon, parathyroid hormones, etc. 6. Orphan GPCRs. All other GPCRs without known functions have been classified as orphan GPCRs. The GPCRs have 7 transmembrane alpha helices with the C-terminal end inward and the N-terminal projected outward. They are characterized by 3 extracellular loops L1 to L3 and 3 intracellular loops L1 to L3 all hydrophilic. Each transmembrane domain consists of approximately 20 to 30 hydrophobic amino acid residues and is long enough, approximately 30 nanometers. The extracellular parts of the receptor can be glycosylated. These extracellular loops also contain 2 highly conserved cysteine residues that form disulfide bonds with transmembrane helices to stabilize the receptor structure. The GPCR arranges itself into a tertiary structure resembling a barrel, with a set of transmembrane helices forming a cavity within the plasma membrane that serves as a ligand binding domain that is often covered by the second extracellular loop. Ligands may also interact with extracellular loops in the N-terminal tail. The N-glycosylation motif in the extracellular part takes part in receptor folding, trafficking, etc. Palmitoylation is the intercellular part after helix is essential for lipid raft localization. The DRY motif in the second intercellular loop takes part in receptor activity and protein-protein interactions. Fig 4.2. Desensitization of the receptor occurs through the 3 motifs: PKC phosphorylation, GRK phosphorylation, and PKA phosphorylation. Figure 4.2 Topological structure of GPCRs. This is a schematic diagram of the structure of an ideal GPCR and the positions of its various alpha helical loops and motifs topologically in the plasma membrane. The side view on the left shows the heptahelical nature with positions of different motifs and chemical groups, while the top view illustrates the relative position of helices. Ligand binding and activation. The ligands for GPCRs range from a photon or subatomic particles to ions H+ and Ca2+ to small organic molecules to peptides and proteins and transmit the resulting signals across the plasma membrane over a distance of approximately 30 A. Small organic molecules generally bind within the transmembrane segments, and peptide hormones and proteins often bind to the amino terminus and extracellular sequences joining the transmembrane domains. High-resolution crystal structures and biochemical studies have revealed that the nucleotide binding pocket of G protein is located between the Ross-like domain and the alpha helical domain of G alpha subunit, surrounded by 4 flexible regions P-loop, switch I, switch 2, and switch 3. The N-terminus of G alpha subunit is reported to be critical for the structure and function of G alpha subunit and is myristoylated or palmitoylated, suggesting the role of this region in the attachment to the plasma membrane. Most studies reveal that the C-terminus of G alpha subunit is inserted into the cytoplasmic pocket surrounded by TM domains with possible interaction with TM6-ICL3. The alpha and beta 1 hinge region of the G alpha subunit also contributes in translating receptor-induced conformational changes to the nucleotide binding pocket as the beta 1 sheet is connected to the P loop that interacts with the phosphate group of GDP. Ligand binding to the GPCR results in the formation of a ligand receptor complex. This results in the interaction of the cytoplasmic third and fourth C3 and C4 loops with the G protein. The release of bound GDP from the inactive G protein is mediated by guanine nucleotide exchange factor GDF. Now binding of GTP in place of GDP is favored by the high cellular concentration relative to affinity. This induces a conformational change in at least two highly conserved segments of the protein, switch I and switch 2, allowing the protein to bind to GTP. In the active state, switch I and switch 2 are bound to the terminal Y phosphate of GTP through interactions with the backbone amide groups of conserved threonine 35 and glycine 60 residues. The ROS-like domain hydrolyzes GTP and provides binding sites for the G beta subunit. However, the proper mechanism of activation of G protein by GPCR upon ligand binding is still under investigation. Different classes of ligands and their examples are tabulated below. Classes ligands. Neurotransmitters epinephrine, norepinephrine, dopamine, 5-hydroxytryptamine, histamine, acetylcholine. Adenosine, opioids. Catechinins, substance P, neurokin A, neuropeptide K. Other peptides, angiotensin 2, arginine vasopressin, oxytocin, VIP, GRP. TRH, PTH, glycoprotein hormones, TSH, FSH, LH, HCG. Arachidonic acid derivatives thromboxane E2. Others, odorants, tastans, endothelins, platelet activating factor, cannabinoids, light. Table 4.1 examples of ligands for GPCRs. Signal amplification. There happen to be a few thousand receptors on the cell membrane of each cell, and on the production of ligand molecules which too is minuscule concerning the response generated, there is always a need for substantial amplification of the ligand responses. Signaling cascades have long been thought to amplify input signals, as each molecule in the cascade can serially activate multiple molecules of a downstream component of the cascade. For example, in the case of the epinephrine hormone signaling cascade, the response needs to be fast to correspond to the flight or fight response. Upon production of epinephrine by the gland, it reaches target tissue with specific receptors. Because the concentration of the hormone in the blood is normally very low, and LT, 10 to 10 m, the signal has to be amplified. Upon ligand binding, a conformational change activates a resting heterotrimeric G protein associated with the inner leaflet of the membrane. Activation of a single receptor can result in even more than 10 active heterotrimeric G proteins, providing a great boost to the transduction of the first messenger and a good deal of amplification of signal. Fig 4.3. The binding of a single ligand stimulates a large number of adenylocyclase, and each of the activated adenylocyclase produces a large number of CAMPs. Now each of the two CAMPs activates a PKA that in turn phosphorylates several other proteins and so on, thus rapidly amplifying the external signal. Figure 4.3 schematic depiction of the amplification of signals by signaling pathways in several orders of messengers. The above chart depicts the hierarchical structure of the CAMP PKA signaling pathway and the distribution of amplifying signals at each level having the ligand epinephrine, adenylocyclase, CAMP, PKA, PKA activated enzymes, and ultimately the products of those enzymes. The concentrations of certain molecules in the cell are mentioned, like the ligand epinephrine is 10 to 10 m, which in turn increases to 10 to 6 m per CAMP. Weak and strong agonists. In simple terms, agonists are chemicals that bind to a specific receptor and induce certain conformational changes to activate the receptor. The agonists and antagonists of GPCRs have been discovered in large numbers, and their application of them has largely been limited to structural studies, including activation of GPCRs and relaying the signal to G proteins. There could be several agonists for a specific receptor and vice versa. In a separate approach, a lot of mimetic synthetic agonists have been made for specific receptors to study their functions in greater detail. GPCRs being a large family, these receptors have an enormous number of agonists. For contra, weaker partial agonists are antagonists who elicit the same response as the agonists but to a lesser extent. These compounds possess low intrinsic activity, thus having partial efficacy in biological responses compared to a strong agonist. Antagonists. Antagonists act opposite to the agonists and reverse the biological response initiated by agonists. GPCRs also have a plethora of different antagonists specific to receptors. Antagonists are either naturally occurring compounds or molecules present in the body or some chemically designed artificial ones with therapeutic significance. Adenylocyclase. Adenylocyclase AC is a protein embedded in the lipid bilayer of the cell membrane. This enzyme catalyzes the conversion of ATP to CAMP, being stimulated by G alpha S subunit. This activation is very essential regarding the broad array of signals that can be transduced by CAMP intracellularly. AC is an integral membrane glycoprotein present as a single polypeptide possessing 12 transmembrane domains. It has an approximate molecular size of 115 to 150 kDa. AC in humans is encoded by 9 different genes and 10 isoforms of this enzyme have been described. In mammals, there are 9 membrane-bound AC1 to 9 and 1 soluble adenylocyclase SAC that generate CAMP based on their distinct regulation and expression patterns, determined by G by subunits, CA2 plus calmodulin, kinases, and NO. For example, G by subunits stimulate AC2 and 4 while suppressing AC1, 5 and 6. CA2 plus calmodulin stimulates AC1, 3 and 8, while inhibiting AC5 and 6. PKC stimulates all acts except AC6, which is inhibited. The membrane-bound enzyme contains 2 similar catalytic domains on the cytosolic phase of the membrane and 2 integral membrane domains, each of which is thought to contain 6 transmembrane alpha helices. Fig 4.4. The binding of a ligand to the stimulatory RS receptor or inhibitory RI receptor results in receptor-mediated activation of G protein, which entails the exchange of GDP by a fresh GTP molecule on alpha subunit and the concomitant dissociation of biconflex from alpha. The alpha subunit of stimulatory G protein has 2 helices turned to switch I and switch 2. 2 regions of the alpha subunit, i.e., G alpha S. GTP, the helix in switch 2 and the alpha 3 beta 5 loop interact simultaneously with a specific active site domain of adenylocyclase, activating it. Activation of it then results in augmented enzyme activity, rapidly converting ATP to CAMP. There is also microheterogeneity of adenylocyclase, although generally, a single enzyme has been assumed to exist for all cyclic receptors. AC1 is important for its role in learning and memory processes, and its activation enhances memory formation. AC3 has a role in peripheral and behavioral anosmia. AC6 overexpression on cardiac cues improves cardiac function. Certain experiments also suggest the bronchodilative and immunomodulatory role of AC9 in the lungs. Soluble AC SAC is mainly expressed in the testes, and any experimental inhibition of SAC causes the inability of the mouse sperm to undergo capacitation. Inhibitors of AX include PMC6, SQ22536, HTSO9836, NKY80, KH7, etc. Forskolin and Calfor syndrome with hydrochloride are known to activate most AX. Figure 4.4 structure of adenylocyclase. This is a pictorial representation of the generalized structure of adenylocyclase, showing the presence of both NH2 and COOH groups on the intracellular side, as well as containing ATP binding, Mg2 plus binding and forskolin binding sites. Receptor tyrosine kinases, RTKs. An enzyme linked receptor or catalytic receptor is a transmembrane receptor where the binding of an extracellular ligand causes enzymatic activity on the intracellular side. Hence these receptors are integral membrane proteins possessing both enzymatic catalytic and receptor functions. The ligands for RTK include insulin, epidermal growth factor EGF, transforming growth factor alpha TGF alpha, platelet-derived growth factor PDGF, fibroblast growth factor FGF, and choline stimulating factor 1 CSF1. Examples of RTK are the ERK family, GDNF, glial cell-derived neurotrophic factor family, NPR natriuretic peptide receptor, TRK neurotropic receptor, and toll-like receptors, etc., among others. The activated RTKs may interact with several types of proteins, including GTPase activating protein, GAP phospholipase CY, and the SRC-like non-receptor protein tyrosine kinases. Figure 4.5 structure and activation of RTKs. The structure of a single RTK is outlined in the diagram. The next 2 insets towards the right show the recruitment of another RTK on ligand binding and subsequent autophosphorylation of tyrosine residues for activation of the receptors. A very common example of the RTK pathway is the insulin signaling pathway. The insulin receptor is an RTK which consists of 2 alpha subunits on the outer face of the plasma membrane and 2 transmembrane beta subunits. The binding of insulin to the alpha subunit triggers a conformational change that allows the autophosphorylation of the tyrosine residues in the COOH domains of the beta subunits. Multidocking proteins like insulin receptor substrate 1 IRS1 then binds one of the phosphotyrosine residues via PTB domain. The activated RTK then phosphorylates the bound docking protein, forming some phosphotyrosine residues, which in turn will serve to recruit SH2 containing signaling proteins, thus amplifying the signal at the receptor level. Pi3 kinase is an enzyme bound to the inner leaflet of the plasma membrane, having 9 homologs encoded by the human genome. PI3 kinase is made up of two proteins named P85 with an SH2 phosphotyrosine binding domain and P110 subunit with catalytic activity. This recruitment of PI3 kinase to the plasma membrane brings it near the sphenonucleotide substrates, leading to the formation of PI3, 4-bisphosphate or PI3, 4,5-triphosphate. Now AKT or protein kinase B, PKB, is a ester-specific protein kinase that plays a key role in multiple cellular processes. AKT possesses a plextrin homology pH domain that binds to the 3-phosphate group of PI3, 4-bisphosphate. The PTK1 kinase also binds to the 3-phosphate groups to get activated. The other two domains of AKT are kinase and regulatory domains, where PTK1 phosphorylates a threonine 308 residue in the kinase domain, and PTK2 inhibits the same to a serine 473 residue in the regulatory domain to render the protein to be fully active. Activation of AKT takes 5 to 10 minutes, but its effects last as long as several hours. In case of insulin signaling, AKT phosphorylates RGC2, ROL gap complex, and AS160 AKT substrate of 160 kDa in the GSP, which is required in the insulin-mediated translocation of the glucose transporter blood 4 to the plasma membrane. Besides increasing the exocytosis of blood 4, AS160 inhibits its endocytosis via downstream target RAP10 and adipocytes. This facilitates the uptake of glucose from the blood, thereby bringing about the hypoglycemic effect. Figure 4.6. Figure 4.6 RTK signaling with the insulin hormone is ligand. This diagram depicts the exact signaling interplay between RTK pathway members that takes place in response to insulin culminating in the localization of blood 4 transporters to the cell membrane. When insulin molecules bind to the RTKs, there occur phosphorylations in the intracellular domain of the receptor, where the insulin receptor substrate IRS binds. IRS facilitates the recruitment of PI3 kinase in close vicinity to PIP2, which is converted to PIP3 by PI3 kinase. Now with the help of two different enzymes, PTK1 and PTK2, AKT or protein kinase B is activated in proximity with PIP3. AKT next phosphorylates certain components of the blood 4 vesicle, causing translocation of the vesicles to the cell membrane, thus facilitating glucose uptake by them. Signaling pathways. AKT signaling pathway. Introduction. Since its discovery 30 years ago, from clone versus AKT oncogene after retrovirus transformed with AKT8, many other labs cloned and characterized the 57 kDAS and THR protein kinase, the cellular homolog of versus AKT. Three isoforms of AKT-PTB are conserved in the mammalian genome: AKT1 PKBA, AKT2 PKBB, and AKT3 PKBG. AKT has been identified as a sphenonucleotide 3 kinase PI3K, effector molecule and acts downstream to PI3K, a lipid kinase that acts in response to insulin growth factors. Mechanism and regulation. Class 1 PI3K phosphorylates the inositol group of the sphenonucleotides, resulting in lipid second messengers PTDINS3, 4p2pi3, 4p2, and PTDINS3, 4, 5p3kip3. These serve as a docking site for membrane localization of plextrin homology pH domains containing molecules like AKT and PTK1. Once phosphorylated, AKT regulates various cellular functions downstream, like cell survival, proliferation, metabolism, cellular growth, and many others, by either activating or inhibiting molecules like FOXOs, BAD, GSK3, MDM2, TSC2, caspase 9, AS160, PROS40, and enos by phosphorylation. Phosphorylation of FOXO3 leads to survival signaling, and its overactivation can induce cell death by apoptosis. AKT inhibits GSK3 beta protein by phosphorylating its subunits, resulting in dephosphorylation of glycogen synthase and an increase in glycogen synthesis. Concomitant to GSK3 beta discovery, which is an AKT substrate from insulin-stimulated cells, it led to the finding of hundreds of other AKT substrates associated with various cellular functions and diseases. Physiological significance. AKT signaling pathway has been reportedly observed to influence various disease progression and shown to be a promising therapeutic target as such or in combination, for example, in case of solid tumors. Allosteric antagonist MK2206 has been shown to inhibit AKT kinase activity in a dose-dependent manner in breast, melanoma, and lung cancer. MK2206 showed to have greater inhibition as compared to other ATP-competitive inhibitors due to the fact that allosteric inhibitors can lock kinase activity in close conformation. Using AKT inhibitors as a single therapeutic agent may not be effective in the long term as inhibition of AKT kinase leads to induction and phosphorylation of other receptor tyrosine kinases, which in turn can activate PI3K by some other non-AKT targets downstream. Preclinical data obtained from MK2206 in combination with anti-estrogen therapy showed to be more efficacious in controlling breast cancer cell proliferation. Besides activation by external stimuli, the AKT signaling pathway is also regulated by phosphatase P10, which inhibits AKT activity. P10 tumor suppressor is a prime target of inactivation in somatic cancers. The enzymatic action of P10 exerts an opposing effect of PI3K, and thus leads to the accumulation of PIP3. Figure 4.7 AKT network and downstream signaling for different cellular activities. Once phosphorylated, AKT regulates various cellular functions downstream like cell survival, proliferation, metabolism, cellular growth, and many others by either activating or inhibiting molecules like FOXOs, BAD, GSK3, MDM2, TSC2, caspase 9, AS160, PROS40, and enos by phosphorylating. c-Met signaling pathway introduction. Receptor tyrosine kinase c-Met and its ligand hepatocyte growth factor HGF have been reported to be associated with various types of malignancies. Proto-oncogene c-Met is located on chromosome 7q21. c-Met is expressed in epithelial cells of various organs like kidneys, muscles, pancreas, liver, and bone marrow. HGF secreted by mesenchymal cells has a pleiotropic effect as well as cytokine and influences cellular function by promoting cell survival, proliferation, scattering, motility, and morphogenesis. It also plays a very important protective function in various disease conditions like liver cirrhosis, progressive neuropathies, and so on. Mechanism and regulation. The inner side of the c-Met comprises a tyrosine kinase catalytic domain bordered by juxtamembrane and carboxy terminal sequences. c-Met-mediated cell scattering involves disruption of cell-cell contact by cadherin associated changes leading to cell motility, which is a vital process in embryogenesis and wound repair. HGF binding to CMET results in cascade of events downstream, which initiates from c-Met receptor homodimerization and subsequent phosphorylation of tyrosine residues Y1234 and Y1235 located within the catalytic loop of the kinase. Followed by this, the carboxy terminal tail becomes phosphorylated, which then recruit adaptor proteins or signaling effectors. c-MET can interact with various adaptor proteins, which include growth factor receptor bound protein 2 (GRB2), the effector molecules PI3K, the transcription factor signal transducer and activator of transcription STA3 phospholipases, and many more. Additionally, GRB2 associated binding protein 1 (GAB1) has a distinctive association with c-Met and, once phosphorylated by c-Met, generates various binding sites for other adaptor molecules downstream. Different tyrosine residue phosphorylation influences the type of downstream effector molecule association. For example, Y1313 phosphorylation leads to its association and activation of PI3K, which induces cell proliferation. Similarly, for induction of the mitogen activated protein kinase (MEPK) cascades, c-MET stimulates the Rat sarcoma viral oncogene homolog ROS by binding with GRB2 and SHC. This is followed by a couple of downstream adaptor molecule phosphorylation that eventually triggers the effector kinase MEPK MEK, and finally, MEPK, which is responsible for mediating various cellular functions ranging from cell proliferation, regulation of cell cycle, survival to cellular motility by regulating many genes. Fig 4.8. Figure 4.8 diagrammatic depiction of c-MET signaling pathway representing downstream adapters and mediators. HGF binding to c-MET results in a cascade of events downstream, which initiates from c-Met receptor homodimerization and subsequent phosphorylation of tyrosine residues Y1234 and Y1235 located within the catalytic loop of the kinase. Physiological significance. c-Met regulations were studied a lot in relevance to the development of drug resistance to cancer therapies. The c-Met interacts with various other RTKs to influence cancer cell progression and survival. Activation of EGFR and c-Met in the cells possessing both receptors leads to synergistic regulation of the modulators downstream. This could result in various crosstalks. For example, transactivation of c-Met can be mediated by RON, receptor dorogene receptor in the absence of HGF. There is growing body of evidence suggesting the role of c-Met expression in cancer progression, which could be due to overexpression or constitutive activation of c-Met. This could be due to gene amplification or another type of gene lesion or upregulation of c-Met expression. Overactivation of c-Met is being reported in esophageal, medulloblastoma, colon, and gastric carcinoma, as well as it is also a chromatostatic gene suggesting a potential role of c-Met targeting in cancer. Hypoxia is also known to play a role in the activation of c-Met. HER2 signaling pathway. Introduction. HER2 is a human analog of the oncogene NOI in rodents. The NOI was first discovered in a rat-induced brain tumor model. This gene is homologous to the epidermal growth factor receptor (EGFR) gene. It has been observed that EGFR-related gene gets intensified in the human breast cancer cell line and is known as HER2. There are four members under HER2 family: EGFR, HER1, AIR1, HER2, 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 NOI, HER3, and HER4, AIR4. There is species-specific gene nomenclature for HER2. For example, HER2 is used in the context of a human gene while MOI is used in reference to rodents, and ERK to denote the gene in reference to either species. Mechanism and regulation. This family of proteins belongs to growth factor receptor type 1, which are transmembrane receptors involved in intracellular signaling pathways upon external stimulus. They possess an extracellular binding domain, followed by a transmembrane domain and finally, a tyrosine kinase domain located intracellular. Fig 4.9. Figure 4.9 HER2 MOI structure. The extracellular binding domain consists of two units for ligand binding LD1 and LD2, linked by two cysteine-rich regions CR1 and CR2. This is followed by transmembrane. Intracellularly, there is a tyrosine kinase TK and a cytoplasmic tail CT. Upon association of the extracellular domains with its ligand, HER undergoes dimerization and is followed by transphosphorylation of the intracellular domain. Upon association of the extracellular domains with its ligand, HER undergoes dimerization and is followed by transphosphorylation of the intracellular domain. Consequently, phosphorylated tyrosine residues create various docking sites for other signaling molecules and lead to the activation of surplus downstream signaling pathways. Some can even lead to crosstalk between different transmembrane signaling molecules, as shown in Fig 4.9. Partner determination during dimerization of HER2 is a key determinant of signaling activities downstream. Heterodimerization is favored over homodimerization for signaling activities. Partner selection appears to be a key determinant of signaling activity among her proteins and their signaling functions. Unlike the other protein receptor members of the family, HER2 possesses constitutive active conformation, so it is constitutively active and lacks specific ligand binding activity. For signaling functions, it is dependent on its ligand-bound heterodimeric partners. HER2 family proteins are associated with various cellular processes like cell survival, proliferation, motility, cell cycle regulation, and so on by interacting with downstream signaling kinases. Figure 4.10 schematic representing HER2 family signaling network and effector molecules downstream. Upon association of the extracellular domains with its ligand, HER undergoes dimerization and followed by transphosphorylation of the intracellular tyrosine domain, creates various docking sites for other signaling molecules and leads to activation of surplus downstream signaling pathways. Physiological significance. Phosphorylation of HER2 receptors results in the activation of many other signaling pathways, more importantly, MAPK, PI3K, and PKC protein kinase C. These are the pathways that regulate cellular proliferation, survival, motility, invasion, and angiogenesis. HER2 overexpression is mostly related to the development of breast cancer besides its association with the ovary, endometrium, colon, cervix, uterus, and many other cancers. Its apparent overexpression is due to amplification, constitutive or overexpression of HER2 gene. Around 15 to 30% of breast cancers are associated with HER2 overexpression. HER2 expression has reportedly shown a significant correlation between overall survival and recurrence in breast cancer patients. Hence, HER2 is being studied intensely as a possible cancer therapeutic marker. HER2 has been shown to be associated with the development of resistance to estrogen therapy in HER+ breast cancer patients. HER2 overexpression makes the tumor cells resistant to endocrine therapy, like tamoxifen, by helping cancer cells to proliferate even in the absence of activation of estrogen receptors. NFP signaling pathway introduction. Transcription factor NFP or nuclear factor catalyte chain enhancer of activated B cells family of proteins regulates various important cellular processes and influences genes involved in a plethora of biological activities, which includes functions like adaptive and innate immunity, stress responses, inflammation along with cellular growth and apoptotic responses. Active NFP forms by either homo or heterodimerization of DNA binding proteins from the REL family. Each of these family members comprises a DNA binding region within the REL homology domain as well as a nuclear localization signal. The inhibitor family of proteins is like IKB alpha, IKB beta, IKB gamma, IKB epsilon, and BCL3 regulate NFP activity and make complexes with NFP upon induction for its inactivation or by feedback loop. Fig 4.11. Various cytokines, stress signals, and inflammatory molecules are involved in inducing NFP regulation. Figure 4.11 schematic representing a classic case of a dissociation from NFP complex upon NFP activation. Demonstration showing IKB phosphorylation leads to ubiquitination and protease degradation of the NFP complex protein from P100 to P52. This releases NF-kappa B from its complex with IKB and unmasks the nuclear localization signal. Regulation and mechanism. In a typical NF-kappa B signaling pathway, NFP activation initiates upon dissociation from the complex. As shown in Fig 4.11, an NF-kappa B2P100 REL-containing NFP complexes are inactive in the cytoplasm. Upon reception of a signal from receptors like CD40, BR3, and LT-beta-R, it induces phosphorylation of NF-kappa B2P100 by alpha. This phosphorylation leads to activation of the NF-kappa B complex by proteasomal degradation of the P100 subunit to P52. The NF-kappa B2P52 then translocates to the nucleus and initiates the activation of target genes. Different NFP composition is associated with diverse biological processes. NFP response could be pro-apoptotic or anti-apoptotic depending on the type of cell and induction. In immune cells, it is mostly anti-apoptotic in function and prevents apoptosis of immune cells like B cells or T cells upon induction. It is also involved in the regulation of the TPRO inflammatory signaling pathway and leads to the expression of pro-inflammatory molecules like cytokines or chemokines. NFP responds to pro-inflammatory cytokines like TNF-alpha and IL-1, which plays a very important role in chronic disease pathogenesis like those of asthma, rheumatoid arthritis, inflammatory bowel disease, and other inflammatory diseases. For this reason, NFP has gained much attention in anti-inflammatory research. Changes in the mechanical environment, like changes in cytoskeleton assembly, could change the nuclear localization of NFP, leading to its activation, resulting in increased cellular motility by formation, along with other changes in transcriptional regulation, which plays a very important role in cancer cell motility. NFP has been in focus for therapeutic intervention in anti-cancer development. Along with being associated with tumor cell progression and inducing metastasis, there are reports suggesting the role of this pathway in sustaining cancer stem cell-like property assists in tumorigenesis. Notch signaling pathway introduction. Amongst various other signaling pathways, Notch signaling pathways play a very crucial role in the transmission of information associated with development, like embryogenesis. Notch signaling pathway affects cellular differentiation in a wide spectrum of organisms and has a unique feature of a very simple structure and very few core components. This pathway is named after the receptor, Notch, which is a single membrane spanning multi-subunit protein. Most of it is extracellular, and only part of it is cytoplasmic NCID attached to the Notch receptor. Fig 4.12. This pathway is very important for cell-to-cell communication during cellular development. Mechanism and regulation. Upon ligand binding with Notch releases an enzyme gamma secretase that cleaves NCID, which then gets transported to the nucleus. Upon arriving in the nucleus, it breaks the association of inhibitor protein complexes like NCOR and CSL. This activates the Notch signaling pathway by turning on target genes, and it requires binding with some other proteins like P300 and MEML. Figure 4.12 Notch signaling pathway showing its core components. Upon activation, it requires binding with some other proteins like P300, MEML. It turns on Notch target genes. Physiological significance. Notch signaling pathway induction could result in a proliferative or apoptotic response, depending on the context. In cancer cells, it could act as a tumor suppressor or could induce an oncogenic signal. This is potentially the result of crosstalk between Notch signaling with other pathways. This pathway has long been sought for cancer therapeutic interventions. Mutations in the Notch pathway has been associated with various disease conditions and developmental defects in human, which involves deficiencies in vital organs like the liver, heart, and eye, as well as malformations of the vertebral. Mutation in the Notch1 gene could lead to T-cell acute lymphoblastic leukemia or aortic valve disease, while a mutation in Notch2 could lead to Alagille syndrome, Hyde-Cheney syndrome, osteoporosis results out of severe bone resorption, and so on. P53 signaling pathway. Introduction. Multifunctional transcriptional factor P53 regulates more than 2,000 gene activities. P53 has major impacts on maintaining genomic stability along with fidelity, many cellular processes, metabolism, cell longevity, and is a tumor suppressor. It gets activated as a stress response which could be genotoxic stress, hypoxia, oncogenic trauma, and heat shock. P53 can block cell cycle progression and cellular growth by putting cell cycle arrest and assist cells in enabling them to recover from DNA damage or advancing to cellular death if the stress insult is very detrimental to the cell. P53 mutation has been associated with more than 50% of cancers in humans and hence has been a very promising and potent biological target in cancer drug development. Figure 4.13 diagrammatic depiction of P53 signaling pathway, its induction and effective roles downstream. P53 plays a very crucial role in synchronizing a complex network of signaling pathways that suppresses the proliferation of any aberrant cell. Mechanism and regulation. Under normal circumstances, P53 expression is inhibited by two proteins, namely MDM2 and MDMX. MDM2 initiates P53 proteasomal degradation by attaching ubiquitin of molecules to P53 by E3 ligase function. MDMX regulates P53 function by suppressing its transcriptional activity. Though its activity is independent of MDM2, it influences MDM2 associated P53 degradation. P53 regulates MDM2 levels by a feedback loop, while MDMX levels are regulated by MDM2 through ubiquitination.
These inhibitory proteins play a very critical role in the regulation of p53 functions upon exposure to stress conditions like ultraviolet radiation and ionizing radiation, which are mainly DNA-damaging agents, results in inducing activity of several kinases like ATM, checkpoint kinases, and ETR, which modifies the association of p53 with MDM to an MDMX big 4.13. Consequently, it results in conformational changes in these proteins, leading to p53 steadiness. Once activated, p53 influences the transactivation of various other target genes. Depending on the type of stress or damage caused to the cell, the p53 transactivation of the target gene could be for DNA repair, apoptosis, and cell cycle arrest, or could be senescence, irreversible cell cycle arrest. p53 can also interact with the BCL2 family of proteins and MOMP, mitochondrial membrane, to induce mitochondrial outer membrane permeabilization, which eventually leads to the release of cytochrome C followed by caspase activation for the apoptosis to take place. Metabolic pathways can also be regulated by p53 in tumor cells by inducing enhanced respiratory functions like oxidative phosphorylation, which suppresses tumor-feeding glycolysis. Physiological significance. p53 gets induced under stress conditions which could be due to genotoxic agents, radiations like UV rays, gamma rays, or other oxidative stress. Its activation could lead to cell cycle arrest, providing stress cells to recover from the damage or induction for apoptosis, eliminating severely damaged cells. Under low stress conditions, p53 expression is very low, leading to its degradation mediated through MDM2 by proteasomal degradation. This negative feedback mechanism keeps a check on p53 levels under non-stressful conditions. It also has pro-survival functions and assists in maintaining cellular homeostasis functions by adjusting the status of nutrients, growth factors, and hormones. The switch to aerobic glycolysis from oxidative phosphorylation is a prominent characteristic of cancer cells. p53 mutation in cancer cells leads to uncontrolled cancer cell proliferation without any restriction for cell cycle arrest or apoptosis, even after the occurrence of genetic damage in the cell. For this and many other factors, p53 has been a prime therapeutic target in cancer research. ROS-RAF-MEK signaling pathway introduction. Activated receptors frequently transmit signals through intercellular signaling proteins called kinases. These kinases add a phosphate group from ATP onto a substrate protein called phosphorylation. Kinases are often themselves activated by other kinases via phosphorylation and can organize into phosphorylation cascades. One such important class of phosphorylation cascade is called the mitogen-activated protein kinase (MAPK) cascade Fig 4.14. The ROS-RAF-MEK signaling pathway is a typical MAPK signaling pathway implicated in external stimulus-mediated cell growth, differentiation, and cellular death. The external stimulus could be a growth factor, inflammatory cytokines, or could be environmental stress. This cascade of signals combines signals from cell surface receptors to regulate gene expression by acting on transcription factors. Depending on the type of stimulus obtained from cell surface receptors, the fate of downstream signaling is decided, which could be from cellular proliferation to induction of apoptosis. Figure 4.14 phosphorylation cascade. Phosphorylation of upstream kinases leads to make phosphorylation of consecutive downstream kinases. Mechanism and regulation. The cascade initiates with ROS activation, which is a GTP binding protein upstream of many signaling pathways like RAF, MEK, ERK, RELEF-RAL, and PI3K, AKT. ROS has many subtypes broadly: HA-ROS, P-ROS. Their effect on mediating different pathways is also varied. For example, P-ROS has stronger control in regulating MEK, ERK as compared to HA-ROS, whereas HA-ROS is involved more in PI3K, AKT pathway as compared to P-ROS. ROS activation initiates with ligand binding with the receptor tyrosine kinase, which leads to its phosphorylation, creating a docking site for adaptor proteins GRB2, SH2, which activates ROS activating protein. Receptivating protein, SOS is a guanine nucleotide exchange factor, GEF, SOS GRB2 complex, SOS pro residues bind GRB2SH3. ROS activating protein phosphorylates an active ROS protein with the help of GTP molecule and hence leads to ROS activation. ROS activation activates various MEPK pathways and distinct biological responses, Fig 4.15. The signal is transmitted to the nucleus through activation phosphorylation of multiple transcription factors. ERK1/2 also translocates to the nucleus upon stimulation. Phosphorylation of nuclear translocation sequence (NTS) within the kinase domain allows interaction with nuclear importing proteins via nuclear pores. Figure 4.15 diagrammatic depiction of ROS activation. ROS activation initiates with ligand binding with the receptor tyrosine kinase leads to its phosphorylation, creating a docking site for adaptor proteins GRB2, SH2, which activates ROS activating protein. Protein kinase family members RAF, RAF1, BRAF, and ARAF are downstream to ROS and are also referred to as ROS effectors. They activate MEK pathway downstream by protein phosphorylation and are involved in cell death regulation. Physiological significance. Downstream to RAF is MEK1 and MEK2 protein kinases associated with the ROS-RAF-MEK signaling cascade, which regulates a plethora of biological activities like cell proliferation, metabolism, differentiation, migration, cell cycle progression, and apoptosis. MEK1 is dual specificity protein kinases and is activated by phosphorylation by RAF kinases. Figure 4.16 signals activate distinct MEPK pathways and biological responses. Figure demonstrating crosstalk between different pathways. Downstream to RAF is MEK1 and MEK2 protein kinases associated with ROS-RAF-MEK signaling cascade, which regulates plethora of biological activities. MEPK is involved in the integration of multiple pathways, resulting in the convergence of transcription factors by translocation to the nucleus. It has subcellular compartmentalization and is involved in subcellular crosstalk with other pathways that modulate MEPK signaling. The duration and amplitude of MEPK phosphorylation depend on the degree of specificity. For example, NGF, sustained activation via RAF1, BRAF differentiation, EGF, transient activation via ROS-RAF1 proliferation. MEPKs can be sequestered at the nuclear periphery by lamins or recruited onto chromatin to regulate transcription by phosphorylating transcription factors, recruiting co-regulators to DNA, phosphorylating histones to alter chromatin structure, and binding specific DNA sites. ERK1/2, involved in regulating apoptosis and cell proliferation, plays a very important role in regulating ROS-RAF, MEK signaling pathway, activated by RAF. They belong to the serine threonine kinase family and are activated by kinase MEK1/2 dual specificity. ERK1/2 have almost 160 downstream targets, which it regulates by phosphorylation and involves many different transcription factors like NF-kappa B, CJUN, and CMYC. Mutations in the MEPK signaling pathway are associated with various cancers. RTKs have usually amplified while ROS-RAF is mutated, and MEK1/2 and ERK1/2 are hyperactivated. The mutation frequency of ROS subtypes is diverse in different cancers, with P-ROS being the one with the highest mutation frequencies. These ROS possess the abnormal capability to activate RAF, MEK, ERK, and PI3K, AKT cascades. Noonan syndrome is an autosomal dominant disorder associated with multiple developmental abnormalities in some cancers. Related to gain of function mutations in tyrosine phosphatase -2 St. Helena pounds per PTP and 11. SHP2 mutants prolong signal flux through ligand-dependent ROS, MEK, ERK, leading to increased proliferation. JAK-STAT signaling pathway introduction. JAK-STAT or Janus kinase signal transducers and activators of transcription is an important signaling pathway that responds to a range of growth factors and cytokines and hence translates the extracellular signal into transcriptional activation. This pathway is involved in transducing a multitude of signals in almost all multicellular organisms and helps maintain homeostasis, immunological development, and other developmental functions. The pathway of signaling involves JAK activation induces cellular growth, migration, differentiation, and cellular death by apoptosis. Mechanism and regulation. As mentioned above, JAK-STAT pathway gets activated by a range of ligands. They get activated either extracellularly upon ligand binding or intracellularly and eventually lead to either homodimerization, e.g. growth hormone, or heterodimerization, e.g. interleukins, of receptor subunits. This multimerization of receptor subunits transduces the signal to the JAK kinase, which is associated with cytoplasmic domains of the receptor subunits. There are 4 members in JAK family: JAK1, JAK2, JAK3, and TYK2. And C-terminus comprises of tandem kinase homologous domain. Upon receptor dimerization, 2 JAK subunit comes closer, which assists in transphosphorylation. Subsequently, activated YOX transduce signals further downstream by phosphorylating additional targets, majorly including the substrate STATs, Fig 4.17. There are 7 members in the transcription factor STAT family that bear a conserved tyrosine residue near C-terminus. Once this tyrosine residue is phosphorylated by JAK, this leads to the dimerization of STATs. Once phosphorylated, the STAT enters the nucleus and binds to its target genes to activate to suppress its activity. Besides basic components of simplified JAK-STAT pathways, other effector molecules interact with the pathways. Some adaptor proteins like MYC are STAT-interacting proteins and induce JAK-STAT activation. There are 3 broadly classified negative regulators of the pathway, namely signaling suppressors like SOCS, suppressors of cytokine signaling, inhibitors like PIAS, protein inhibitors of activated STATs, and phosphatases like PTPs, protein tyrosine phosphatases. Leptin is a 16-kDa hormone released by adipocytes and is in proportion to the stored fat level. The OR leptin receptors belong to the class 1 cytokine receptor family and act through JAK-STAT signaling pathway. Leptin is present ubiquitously in most of the tissues, plays a role in immunity, angiogenesis, wound healing, metabolic pathways, and bone and cardiovascular functions. It plays a pleiotropic role in crosstalks with many signaling pathways like PI3 kinase, MEPK, etc. Physiological significance. As discussed above, JAK-STAT pathway plays a very crucial role in regulating a plethora of downstream signaling by translating extracellular signals as a response to ligand binding to a specific JAK receptor complex.
The mutation which loses regulation in the pathway has been associated with various disease conditions, which could be inflammatory diseases, erythrocytosis to certain cancers, like in the case of leukemias. Loss of function mutations in JAK3 resulting in loss of T and NK cells has been associated with severe combined immunodeficiency disease (SCID). Gain of function mutations like point mutation, insertion, and deletion has been reportedly associated with various malignancies. Figure 4.17 JAK STAT pathway activation and downstream signal transduction which leads to the action of dimerized STATs on its target genes in the nucleus. WNT signaling pathway. Introduction. WNTs, wingless related integration site, signaling pathway composed of WNTs, which are secreted factors, plays a very crucial role in multicellular organism development. These secreted factors bind to frizzled receptors which lead to the